Hello grade 12s. So for this lesson we will be starting with unit 2, meiosis, and this lesson will focus on a bit of introductory information and then the process of mitosis. So chromosomes can be found in the nucleus of all living cells, and we know this from grade 10. So when the cell is not dividing, chromosomes appear as a chromatin network, which is this thread-like structure. And when the cells divide, the chromatins appears as chromosomes. So the chromatin network will then condense, shorten and thicken to form these chromosome structures. Okay? So when it is not dividing, chromatin network. When it is dividing, chromosomes. Before the cell divides, the genetic material must be duplicated. And we know this from DNA replication, right? And this takes place in interphase. Okay, so it ensures that both daughter cells formed have identical genetic information to that of the mother cell, and then obviously to each other. So you will remember this picture from DNA replication that we already did, but you have two chromatids that are your sister chromatids that are identical, connected by the centromere. Chromosomes. So somatic cells is what we call the body cells, and the word gamete refers to the sex cells. Okay, so somatic cells in a species contains the same number of chromosomes, and this is true for all the body cells throughout your body. They all have the same number of chromosomes except for your gametes or your sex cells. Okay, so sexually reproducing organisms have an even number of chromosomes. We call this the diploid chromosome number, or 2n. Okay, and this is because you have one set from your mother and one set from your father that form homologous chromosome pairs, which are similar in size, shape and genetic composition. So your somatic cells, again, we call this 2n, the diploid, and then your gametes or your sex cells are n, haploid. So when fertilization of the egg, N, and sperm, N, occurs, the zygote will form if these two fuse, okay? So the zygote will then be 2N, because this and that came together, fused to form the zygote. So N plus N is 2N, which is now again your diploid. So the zygote will then divide by mitosis and then forms an embryo which consists of many cells, and obviously this will become the baby. So in humans, the mother's egg has 23 chromosomes, which is your haploid. The father's sperm has 23 chromosomes, again haploid, and this will form the zygote, 46 chromosomes, diploid. Okay. So what is a karyotype? A karyotype is basically just an image with the whole diploid set of chromosomes that are arranged based on their size, shape and number within a somatic cell. Okay, so it's the chromosome content of a somatic cell and the whole diploid set of chromosomes are then arranged based on their size, shape and number. Okay. So you will see that this is a human cell's karyotype. So in humans, we have 22 pairs of autosomes, or 44 chromosomes, right? So you can see here 22. And then we have one pair of gonosomes, or sex chromosomes, which in males is XY, and in females it's XX. So you won't have both of these. You have either number 23XX or number 23XY, depending if you're male or female. And then that makes a total of 46 chromosomes, or 23 pairs of chromosomes. So cell division. During the cell cycle, a cell grows, replicates genetic material and divides. So the cell cycle is divided into the interphase and the dividing phase. And interphase occurs between two dividing phases. Okay. During interphase, the chromatin network is visible, and cell growth, replication of DNA, and duplication of the centrosome takes place. So here you can see the centrosome is duplicated, our chromatin network is visible, and we know that DNA will replicate during this phase in order to be ready to divide in the next dividing phase. Okay, so during the dividing phase, the chromosomes and nucleus divides. So when the nucleus divides, we call it karyokinesis. Then the cytoplasm, organelles, and cell membranes will divide. Cytoplasm that's dividing is called cytokinesis. Okay? So let's look at what mitosis is. Mitosis is the division of somatic cells, and it produces cells for growth, repair, and asexual reproduction. So it's very important that you note here that it's not for sex cells, it is to produce cells for growth, repair, and asexual reproduction. So producing normal somatic cells. Two daughter cells with the same number of chromosomes as the mother cell are produced, and the mother cell and two daughter cells are identical. So in mitosis, we have 4 phases, and these are prophase, metaphase, anaphase, and telophase. So let's look at the process of mitosis. So in prophase, the threads of the chromatin network will shorten and thicken to become visible chromosomes that we can see here. And of course we know that these chromosomes have the chromatid and the centromere that's in the center. Okay. Then the nuclear membrane and nucleus will disappear. So you can see there's no, this nuclear membrane and nucleus is not present. They disappeared. And the spindle fiber will form between the centrioles to form the spindle. And the centrioles are here because they duplicated in interphase, remember? And now they moved to the 2 opposite sides, to the 2 opposite poles of the cell. During interphase, the DNA was replicated, so chromosomes have 2 identical chromatids that we can see there. And that is it for prophase. Okay. Then we move on to metaphase, which is your next section. So you can remember prophase, pro meaning before, metaphase. The chromosomes are in a single row on the equator, so they're in the middle of the cell. So the chromosomes are in a single row on the equator, and the spindle fibers attach to the centromeres over here. Okay? That is metaphase. Then we move on to anaphase. So during anaphase, the chromatids are pulled to opposite sides of the cell. So ana, opposite sides, okay? So during this phase, the centromeres of each chromosome will divide into 2. As you can see here, we have 2 centromeres on each of the chromatids, so they're divided into 2. And the sister chromatids will separate and move to opposite poles. And then we refer to these as daughter chromosomes as well. These separated sister chromatids, because remember it's now not the whole chromosome, it's just that section, the chromatid. And then cytokinesis will also occur. And cytokinesis is basically the division of cytoplasm, and this will begin here. Okay. Then in telophase, this is our last phase. I always remember this as like them splitting and needing a telephone to be able to communicate. Okay, so telophase. So the chromatids will position themselves at the poles. As you can see here, they move to the poles. The spindle fibers will disappear, and a nuclear membrane is then formed around both of these like little sections of chromatids. Okay, and inside both of these a nucleolus will also form. Okay. Then the cytoplasm will start dividing. This is what we call cytokinesis, remember? And then we are left with 2 identical daughter cells that are now formed. Okay, so that is basically the process of mitosis, and in the next section we will talk more about meiosis.